So we are live on YouTube. I have shared the link.
Hello, Shashank. Please make Farah Shakes your co-host.
Sure. Farasa, you are co-host now. Thank you. I have already made him.
Okay, thank you. Thank you.
Let me know when I should start the presentation.
Yes, sir. Just I will make a brief introduction of you.
Okay.
Thank you very much, sir.
No problem.
Hello, everyone. Good morning to all. And now we are in process of starting a next day session of our 2 days international seminar at MLT College Saharsa. The second day session will start by a wonderful lecture by our renowned resource person, Dr. Farah Shakes. I will give a brief introduction of him before starting the lecture. Dr. Farah Shakes is a postdoctoral fellow in a molecular modeling and computer-aided drug design. He has 2 years experience as computational chemist after master degree at Center of Excellence in Drug Discovery, Rajkot, Gujarat. He has experience in CADD tools and methodologies such as molecular docking, high throughput virtual screening, molecular dynamic simulations, homology, and so on. He, up to now, he has 14 research publications and a long back research track record. So with these words, now I invite Dr. Farah Sheikh to present his lecture. Sir, please.
Thank you. Thank you for inviting me, and thank you to the Prakash Jha and everyone who invited me for this conference. I hope I'm audible to everybody, right? I'm audible to everybody?
Yes, sir, you are.
Okay, good. So we will talk about my field, which I am right now. I am a CAD scientist. I just joined as a scientist position in just 2 days ago at Irish University of Montreal. Now my work is mainly to support the computational chemistry to the ongoing projects happening in the University of Montreal and the Bristol Myers Pharmaceutical Company. So I'll take you to the brief about, should go to the next. We'll talk you the brief about what could be the drug design process. This is a traditional drug design process that you are seeing right now on your slide, where you have a synthetic or natural compound and it goes through the preclinical and clinical trials and it becomes a drug. In the modern days, you have a sophisticated method to identify the targets. And if you don't have a targets, you have a lead compound, then you look from the compound data set itself to come up on some conclusion to find out where these molecules goes and bind to. Once you identify the targets and once you identify the binding site of the molecule, you can further lead optimize and create the SAR, and then it leads to the preclinicals and clinicals. So the CAD has a tremendous role in upbringing the drug discovery processes right now. So as we are progressing, right now what you are seeing on your slide is these two main method of computer-aided drug design is structure-based and the ligand-based. Ligand-based approach is mainly when you don't have a target at all. So you build the QSR model based on the compound library you have. You create the similarities, you generate you take out the molecules and you take out all the properties, and based on the properties and your experimental data, you create a model, and that models will help you to identify future molecules in case of you don't have a protein target to look into it. Once you have a protein target, then you know what could be the binding site of your molecules, and then you study that binding site, and then you look onto the docking methods, and you do a lot of large virtual screening to identify new molecules, and then you select the molecules, and then further you lead optimize the molecule based on the pocket informations, and then it leads to the further process like lead optimization and the preclinical clinicals. So first, when we go into the computational approach, people, and whenever you go to the chemistry department, people have a lot of molecules, but they don't know where these molecules will go and bind. So you must understand that the binding site or at least the target. So in silico base, computational base, so many target prediction methods are available. So it's just a click of button now that you can actually identify a probable or hypothetical target for your molecules. And there are several ways to do that. We have a ligand-based approach, we have a docking and simulation approach, we have a different machine learning and AI-based approach, which are now booming. So what I did is I studied all of this. I studied all the target prediction method first to understand that what approaches they use. Some approach just based on the structure information, some use just only ligand information, some use structure and ligand combined method, some use machine learning method. So I come up with an idea that if the person have just a molecule and if he puts in some tools that I build up, and the tools should be automatically identify the target, and the molecule should go and bind to that target and automatically generate the binding interaction fingerprint and should compare with the existing crystal ligand fingerprint, and also build up the AI machine learning models that can predict the activity for that probable molecule. So what we have done, I have created the LICT map. That is a server which we published in Journal of Chemoinformatics, very good journal, right? And the server is freely available to everybody. So what we are doing is you convert your chemical compound into the SMILE format. And that SMILE format goes and generate the binary fingerprints of your molecules, which we call the molecular fingerprints. We use the RDKit, a Python programming and RDKit modules to generate this kind of fingerprints. Fingerprint is like a binary code of your molecular features. So if this fingerprint, your query compound, your own compound, you put in the SMILE format to our server, our tools use this molecule to generate the fingerprint, and then it compares with the available compound data set in the public domain, and it tries to see that where those compound binds, and then it provides you the hypothetical target that your probable molecule can go and bind to. Then I have built up the new session in my tools where your molecules can automatically go and bind to that particular target, and you generate the docking pose for your target and your molecules. Then we have something called interaction fingerprints generation. So it will generate the fingerprints, how your molecule binds to that target. And then it compares with how this crystal ligand is binding to that target. And then you have a probable understanding that how close you are to the existing experimentally available molecules and its binding pattern. We also have artificial intelligence-based activity prediction method models built up for the 17 different class, ligase, kinase, and different diseases like tuberculosis, diabetes, cholera. So for that, your molecule will go into those models and the selected target class models, and then it will predict the activity, probable activity for your molecule. So it is a complete package, as a web server, complete web server package that provide you the whole results for your molecules. So how did we actually set up everything? This is just an example that how we can use the server which I have built up. This is the one of the paper published in SCA's MedChem about the HIV molecules. So what we did is this 2019 paper, we take out all the molecules which were reported in that paper, and we try to predict with our own LICT map server, and then try to see that whether we can predict the accurately experimental value also for the molecule, as well as the binding pattern of the molecules. As you can see, we are predicting R is equal to almost 0.83 for the 10 compound that they have proposed. So we are very good with HIV prediction models in terms of the docking, interaction, as well as the actual activity predictions. So if somebody is working on HIV, for them also our server will be very helpful. If you go back and see how many we have published this into Journal of Chemoinformatics. There have been more than 25,000 users for just 1 year. I would like to show you this this web server so that and you guys can help. You can see my web server, right? The images which I'm showing you. Yeah. So I'll just try to show this Lichty map. It's a method to predict the target. These are the different class of the target we have. And here you can select the docking methods also automatically. And if you look at the results, once you process, like you can select your molecule here. You can put the molecule here. You can select the class of the target you want to, and then you can you put your email address and everything, and you can click here and put a submit. Once you do the submit, you process to the results section. 1 minute, I have to change it. Okay. Yeah, once you go to that, you process to the results section where you can identify your target list. You can see the docking pose and everything. So I would encourage people to go and try and check this web server. It's completely free, and you even for BSc and MSc student can also do some sort of small project for their academic semester using this particular web server. So, and more details can be found in this publication. So this is the ligand-based approach where you don't have a target, but you have your compound, you have a you have done chemistry and you have a molecule, but you don't know where this molecule will go and do something. Then you can use this method. I'll give you some example of the structure-based method when you already have a structure, protein structure available with you. So how you can come up with the real molecules if you have just the structure molecules. So we have 3 different target here. One is the Ebola virus. Ebola, you know, is very deadly disease, happen in West Africa. It is, the mortality rate is almost 50%. And it's a very deadly disease. So I was invited in University of Oxford for a 10-month of research to identify a molecule for the Ebola Glycoprotein. And I will share the study which we published in Journal of Medicinal Chemistry. And there is another target we are looking is TNFR2, which is also a cancer target. So here the approach is different, where you are targeting a protein binding site. So I'll also explain you this, so you will understand how to target protein binding site. In case of protein binding site is your target aim of the target. And the third is GPCR, which are very famous. And this GPCR is C5AR, chemokine receptor. We had a collaboration with Pittsburgh University, and we identified the first positive allosteric modulator for the target C5AR. So let's discuss about the first Ebola glycoprotein. So before I went to the Oxford University, they said that they identify ibuprofen and turmifene, which are the cancer drug that bind to the Ebola glycoprotein pocket. So when I went there, they already identify a structural, they already identify pocket, they identify a known molecule that bind to. So my task was to identify, using the structural information and the known compound information, my task was to identify novel molecules that can actually block the Ebola glycoprotein replications by targeting this pocket and destabilizing the whole trimer structure. So first I studied the known compound and how they bind in computationally. So we dock all those known compounds that are reported to bind to that pocket, and we generated the scoring pattern for that, and we tried to correlate with the experimental value and try to see which particular scoring method will be very appropriate to identify a new molecule using the large vertical scanning for this particular target. We could see that the specific to the QPLD method, that is a quantum polarized ligand docking, QMM-based ligand polarized ligand docking is identified with very close to the experiment. So we try to use that method as the advanced method after we complete the virtual screening. So we have run a large 2.5 millions of the Chinese medicinal compound, and we have employed that into the Schrodinger, a very standard ways of doing virtual screening. But the main task was to easily inspect those molecules into the pocket and how they bind compared to the known compound. Out of the total 16 compound that we screened, we actually purchased the 8 compound to test into real-time experimental. So you can see these are the dog compound, and this is a real crystal we found for those compound. So out of 8, we found 2 molecules that actually destabilized the complete Ebola glycoprotein trimer. And so far till today, we have the strongest molecule that is 50 nanomolar for Ebola glycoprotein. And this is the best Ebola glycoprotein binder available in the market right now, in the publication right now. So if you see this paper we published, and we were in the cover page of Journal of Medicinal Chemistry. So this is kind of thing that you do once you have a structure. So once you have a good structure, you have a good known inhibitor knowledge where it binds and how it binds, you try to use and utilize that information and try to find the new molecule for that. The second approach I'm going to give you is the protein-protein binding sites, because these are the complex approach. Once you have a 2 protein bind and there is not an obvious pocket information is available. So how do you get out this information? So this is the TNF-TNFR2 complex. So what we did is we done a simulation on the protein and protein 2. And after the simulation, we pull the both protein apart and try to see which particular residues are binding more tightly to the other part of the protein. And therefore, we come up with this region that they are involved into the interaction pattern. So once you block this region with the small molecules, then there is a chance that you block the protein-protein interaction. So this is the way that you try to see a different approach. And this Ppi approach we identify, we created a hotspot, and then we run the virtue scanning using the same method which I explained you in the last Ebola glycoprotein method, except that we included the MD simulation and the MMPPSA calculation, which actually to understand whether these binders are stable in the pocket or not. Because once you identify some molecule, they should compete with the highly charged van der Waals interaction of protein. So that we actually come up with some molecules, and we publish that into Frontier of Immunology. And we also tested this molecule against the TNFR2. They were very weak binders though, but still they could actually inhibit the PPI interactions. The third project which we did is with the collaboration of Pittsburgh University. So this one was the GPCR. This one was the C5AR molecule, chemokine. It's in the cancer, it's in the neuropathy, and obesity and different diseases. So they had a one molecule, but the Pittsburgh University Professor Chang has published a paper with the avacopan, which is the nanomolar binder of the C5AR. And my role was to identify something very similar to it using the structure-based the virtue scanning. So what we did is we perform the same method which I shown you, because once you have a structure, either you do a direct virtue scanning or the QPLD calculation, or you do MD simulation. In case of this, I also predicted the homology model for C5AR, because the initial structure RMS, the resolution was not good. So we tried to see that whether we could predict the very good, with the exact resolution, we could try to see that the proper density and try to create that. Once we run the virtue screening, we tested with their experiment, and we identified that we actually found a molecule that activate the C5AR. It's like a positive allosteric modulator. So this particular work, because the PAM is very interesting, positive allosteric modulator is very interesting for pharma. And if we find some similarity of the target of C5AR to the other target, then we could actually use this molecule as to activate those receptors which are not activated or which are deactivated due to the diseases, in particular to the GPCR. So we identify a series of compounds that are a positive allosteric modulator, and the work is still going on because we must get the nanomolar binder in order to get the crystal structure with this. So we have almost 7 compound with this target. So this is a structure-based approach. So these are the basic thing I explained, how the ligand-based approach work and how the structure-based approach work. My current company project, I am working on breast cancer. I am working on the DEL library creation, DNA encoded library creation. I'm working on the machine learning and deep learning models. But due to this, because they are bound to the pharmaceutical project, so I could not share with you guys. But these are my PhD work, and we have published most of this work. So I could actually explain to you and just to show you that once you have a compound, then you do this. Once you have a structure, you utilize and do the structure-based approach. I'm not sure if the audience understood very well, and also my voice, because it's 1:30 a.m. in Canada right now, almost in the middle of the night. I am presenting with you guys. So thank you, Prakash Jha, and everybody for inviting me and delivering a short talk for people. I would be very happy if you have some question regarding the server. You could go and use the server. You can ask your chemistry student to utilize that server and do some sort of project and help them to write some good papers. Thank you. Thank you. Hello. 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 I have completed this session.
Thank you very much. Thank you very much, sir, for your wonderful lecture. Actually, it was a connection problem here, so we were not able to connect. Am I audible to everyone?
Yeah.
Yeah.
Sir,
I'm Dr. V.D. Tripathi.
Yes.
I have I want to know your comment on a particular statement. Yes, I am a typical synthetic medicinal chemist. I design a small molecule for different targets. But during my journey for last 10 years for synthetic chemistry, applying for designing the molecule for against various targets.
Yeah.
So, in your opinion, how can I utilize the computational aided drug design to design my molecule for a small target up to fullest length? That I used it, but many times what I do when I find my molecule is active against some targets, then I go to check the molecular modeling and docking whether my molecule is binding with the target sites or not.
Yeah.
But this must be done before doing the biological test against some targets. So
sometime you have affordability to do the biological testing and not the skill labor to do the CAD. So what happens is in India, mostly when I was in India, people do have a biological collaboration often, like, you know, they know people doing the particular assay. So they just send their molecules to there. And then once the molecule hits, they go back and see the target and generate some interaction profile. So there is no harm in getting the interaction profile of your chemical compound. But the good study is sometimes that you don't have a target at all. So therefore, I share the web server which I was showing to you. This web server particularly is very helpful because you can actually put your molecules in SMILE format here. And before doing the assay, you try to identify where this molecule can go and bind to. So like you can just select one of the target. You can put your name and you can put your email address and just submit it. So it will rerun. It will run and it will provide the complete detail of your molecules. And then you select the target and you find a collaborator to do the assay. Yes, that is how it should be done. But in our case, mostly people have the, what do you say, everything, the biological setup, everything. So you just directly send your molecule to the biology. And then later you try to see the interaction pattern of your molecule. But I think both ways okay. But once you have some strong collaborators which actually do a, what do you say, binding assay, the real-time binding assay, and then you try to compare your molecule binding IC50 to your, the experimental IC50 to predicted scoring, and then you generate the correlation. So what happened there is you actually build up the SAR, then you can actually lead optimize very well. So selecting assay is also very important in terms of, going forward with your particular moieties. We just recently published in SCS about the dihydropyridine glycoprotein inhibitors in just few days ago, where we actually designed the molecule based on the pocket. And then we run the binding assay, specifically PGB inhibin assay, and then we correlate the binding IC50 with the docking score. So then we understand, okay, if we are modifying this, then the scoring is happening, modification here, then the activity is losing or activity is gaining. So this is called a SAR buildup, which is helpful if we have a good CAD initially applied, and then you design, and then you go to the assay and you get a good results. So this is just a comment from me.
Thank you. Thank you very much, sir, for a wonderful lecture. So now let us thank Dr. Farah Shakes on the behalf of MLT College and this organizing team. I convey my heartfelt thanks to you for delivering this wonderful lecture, and I hope we will have a great relationship with you in future more. Thank you very much, sir.
Thank you. Bye-bye.
Now we will move forward to for our next session. Our next two speakers will speak offline, and we will start very soon. Thank you.
I traveled by a new way from Biraul to Saharsa via Kosi, via the yes, via the bridge on Kosi for the first time. I think the road came into existence perhaps 5 years back, some 5 years back. But it was first occasion for me to travel by this road. I still recollect that I did my matriculation from a school, Amadi, near Mordigan, supported by the school, Amadi Mordigan, where my father was a teacher, and it took 24 hours. It took about 24 hours by train journey, taking train journey from Mumbai to Mordigan. 24 hours, changing 4 trains from Mumbai to Manzio, then Saharsa, and again to Saharsa. So it took 24 hours. And I had memorized names of all the stations that were there. So I had become again nostalgic. This college, first I knew from the here, and we heard a great orator. He was an artist at Patna radio, Patna. From there, this program, we heard him several times. So he was a legendary figure at Saharsa College. So my memory was known by the name of Ppi Approach, was a great professor here. And then, as a student of botany, teachers from this college, there used to be a janitor at the college, and Dr. Divisha, Dr. Divisha, he wrote Madhikro, and he was a Samudri, and then Ravi Kumar Singh, and then Dr. Ashok Kumar. So, as a botany, I know the dinner of the botany teachers here, and I had several occasions to come here to this lesson, and then as a janitor in the same department. So, Saharsa College has been to me since our time. So, with this introduction, I would like to come to my topic. Yes. Multi-place in science and technology is the topic of now today's seminar that is being organized. That is being organized here at this college. And handbook. I organize the principal and members of the organizing committee for presenting this assembly. My topic is today we are satisfied and the basis for the development of the art. Hana has come to limelight during COVID time. This age of globalization, in this age, nothing, neither a product nor any person as such of that matter, who does have significant qualifications, significant property, cannot live in for a long time. Same is the case with Makana. It's an aquatic product. Those in our area means starting from only districts of Nepal up to Kishangan and even beyond, which beyond the district. So Makana has got its distribution of cultivated population in 8 to 10 districts of Bihar, India, then we have 3 districts of facilitation, that is Saharsa, Supaul, Adekla, and then 4 districts of Purnia region, Purnia, Katihar, and Kushan. So in all 8 to 10 districts, Makhana is cultivated as a crop. It was not recognized as a crop earlier. Even today, when we go to internet, go to Google, and search about Makhana, what is the original one of which is a product, you will get it available to see it. Those who have not seen this plant with their eyes, they generally take it as a product of lotus. Of course, lotus also belongs to the same family, but this direct payoff is different related to the family. Same family is BSc, from which now a separate family, UASC has been carved out, and Indonesia are two class under this family. So Makhana has come to the limelight during COVID times, and none other than Mr. Modi. He spoke a number of times about the universal property of Makhana. So that just triggered movement, and Makhana came to light all over the world as a booster plan, both particularly during COVID. And this yes. इससे पहले वाला मुश्किल नहीं होगा चलिए so these are the edible forms of makhana. These are the edible forms of makhana as sold in the market. And urea ferox, that is makhana in field condition. This condition you find here in your district also, as to how the leaves, the big leaves, they are falling flat on the water surface. It's closely allied genus is Victoria. Victoria also has its upturned margins even during its maturity. But here, the early leaves, they have their upturned margins, and when grown, when mature, they fall flat on the surface. Makhana crop facing the flood of water runs on the margin have started drying. As you see, the only limiting factor that is required for makhana cultivation, that is limiting factor, that is water. Of course, there are a number of materials that increase the crop, but here water is the most limiting factor. Just see what is the situation. Just water is being added, it is being irrigated, sufficient water should be there. Just see how the plants, they are being collected for transplantation in some other water area. Yes, we have two important research centers of makhana. One is at Darbhanga, that is research center for makhana under ICAR, Indian Council of Agricultural Research Center, and the other is at Purnia, Bhola Paswan Sarvati Agriculture College at Purnia. That is another important center, two important research centers of makhana in our state of Bihar. Attempts are being made to introduce makhana as a crop in the non-traditional waterborne area of both within and outside the state. There is a large area in the nation in districts like Gopalganj, Sivan, Jabalpur, Vaishali, etc. The crop has been introduced in the state like UP, Chhattisgarh, etc. Introduction to new areas could be done either through seeds or through transplantation. Fishing community of utilization could be higher during local makhana production, in its cultivation, harvest, and post-harvest operation. As I have already told you, that 8 to 10 districts of Mithila, they are the normal place of its cultivation, but since there is much stress upon raising its production as well as productivity. So the new wonders of the area in other non-traditional districts, they are also being tipped for a plantation of makhana. Initially, there would be some difficulty, but even then, the government has prioritized this area, and so we expect an increase in the production area as well as in the productivity of the crop. Next one. Makhana garments are becoming popular. Wedding garments in cities outside Bihar. Makhana pops are becoming construed as a product of seeds of the full speech to the air. That is untrue. One sequence that makhana, one of the Fungicidal, that is 5 auspicious dry fruits, is product of aquatic crop. It is called makhana is included under Panchameva, that is 5 auspicious dry fruits that are used under holy occasions, sacred occasions. So makhana is one of them. Yes, makhana is gluten-free, which is preferred by those suffering from gluten. And then recently, a study in Pakistan has thrown sufficient light on its hypoglycemic property, and a compound HBAC, hydroxybutyrate acid, beta-caprylate, has been recently reported from this plant as an anti-diabetic compound. Its hypoglycemic property has been proved very recently in an elaborate study made in Pakistan, and the compound of anti-diabetic compound also that has been identified as HBAC, hydroxybutyrate acid, beta-caprylate. So I term it as a nutraceutical, just like soybean. Soybean is held as a nutraceutical. It has got nutritional properties, as well as it is used in as medicine also. Similarly, makhana also could be termed as a significant nutraceutical, and it is full of nutritional property as well as medicinal property. I would elaborate some more detail later. Let's see. Here in our area, it is a cultivated crop, and we know only the pops. We identify only the pops as edible makhana, but in area outside our area, that is in Manipur, its wild populations are found throughout in states throughout Himalaya, starting from the northwestern lakes of Kashmir to the lakes of Manipur lakes. That is famous lake of Manipur, Loktak Lake. I had an occasion to go there and see makhana plant growing there. There in Manipur, they use the non-pop parts, vegetative parts of makhana for making several pieces. You see, Hanji, there is called as Hanji. A number of delicacies they are prepared from this makhana seeds here. Then this is salad. Also eat there. Salad, part of Wisan called salad. Then I have an occasion to welcome an Israeli scientist from Diffusion Technique, Israel. She came all the way from Israel to Darbhanga. At that time, I was at the medicine college as principal, and Israel. She made a visit to Darbhanga in September 2011 to know the details of the cultivation and utilization processes of Gordana. Common English name is Gordana or Fox. Along with another 5% experiments have been discovered from the Victorian side of desert in Israel, along with numbers and angles dating back to the early Middle Pleistocene. So, I showed her slides as to how makhana plantation is done, makhana cultivation is done, and today how is it used. But what was the situation during prehistoric times? From an excavation site, the nuts were discovered, so she got satisfied and she wrote a very good paper that was published in the famous journal Science. So she collected information, verified her information by coming to Darbhanga. You can see Dr. from from Israel. Yes, just see. One single plant of makhana can have at least 10 to 12 leaves at a time. 10 to 12 leaves at a time. Some emerging, some maturing, some decaying. So when we allow unhindered space for the plant to grow, just see. Yes, one single plant of 12 flowers having 7 mature, 2 decaying, and 1 unfolding leaf. That is the situation. But in normal situation, this is not the case. We have at least 4 to 5 leaves only, because a number of other plants are there in the same space, so they have to struggle. But here one single plant is there, and this is a photograph taken at Makhana Research Center by one of my PhD students. So he allowed unhindered space. This is the situation. This is the situation. Yes, the situation of Makhana and Bihar. On the right side, we have the map of Bihar. I have already told you. On the left side, the different states of India, starting from country states of Kashmir up to local Lagos and Manipur. The distribution is there. Of course, that distribution pertains to the wide population. Cultivated population is there only in Bihar and adjoining areas of Bengal. There also will be cultivated. Yes. Why is makhana so important? Which gives us makhana and a unique biology. It has a great food value, it has tremendous nutritional significance. Makhana possesses therapeutic importance. Makhana transforms the curse of water logging into a strategic disease. Possesses biochemical significance and industrial significance. May be used as an effective drug delivery system. Makhana cultivation permits symbiotic growth of certain fishes and releases soil for other crops, including maize, wheat, lentils, etc. And as a social issue, it deals with religious and cultural rituals or lessons from birth to death. Just in figure, this is a photograph from a historic pond, historic tank, got dug by Raja Shiv Singh, under whose patronage was poet Vidyapati, his friend, philosopher, and wife. This big tank is situated in between Ratanpur and Brahmapur villages of Jale district. Those from the Banda district might be knowing the photograph of the white between Ratanpur and Brahmapur villages. Now all these are government ponds, big tanks, got dug centuries ago. But today all of these ponds have become shallower and shallower, and only makhana cultivation is done in these areas. That was not the situation when these ponds, these tanks were in their original shape. They were quite deeper. But today all of them they have become shallower, and this is the situation. This monographic genus has a discontinuous distribution in freshwater pools of northern India, northwest, temperate lakes of Kashmir, and northeast Himalaya, and the states like Bihar, Assam, Odisha, West Bengal, including Bangladesh, Tripura, Manipur. The eastern part, its wide populations are present. Just see, for those who have not seen Victoria, it's closely aligned. It's a big here. Just see how crowded these markets are. It's closely aligned genus, Victoria. It is there in the botanical garden of Kolkata. Yes, then those who have not seen the United plants with their own eye, they will mistake it as a program of lotus. So I have already described it. The third of cultivation. Makhana is cultivated either in water body of 4 to 5 feet depth or in field with 1 feet water. Today, while coming from below to Saharsa, I have seen a number of dozens of makhana ponds, makhana water body, where it is being cultivated under field condition. In Darbhanga and Madhubani, generally it is cultivated under pond condition when there is sufficient water. But even under no requirement, it is being cultivated. Stagnant water system, this is the traditional system of makhana cultivation. Seeds are spread in pond along with some insecticides. Seeds are not required in cold mountain growing ponds. Since that seed of previous crop, some heat of subsequent crop. In some areas, apart from banana, some varieties of fish are also harvested as an additional crop. But it is not likely that fish in Arabia and Kobe. Farmers reported lack of knowledge, practice and vulnerability. So, integrated aquaculture that is practiced in deep water body where makana is grown, and where it is cultivated under field condition, I think that situation is not. Field system is widely practiced in both the districts. In this process, maternal cultivation is carried out in the agricultural fields with depth of 1 to 2 feet. This is comparatively easier in one system, and the recipe can be used for the next field crop. The banana seeds are first grown in pond or any standard water body and then transplanted in the main field. Some farmers are told that they do not grow seeds in pond; they directly sow them in field. This process starts from 3rd week of February and transplanting is done in 2nd week of April. Throughout that process, the transplanting process reduces the entire duration of farming by 2 months. Generally, it starts from January and the cycle goes up to August when makana is harvested by diving into the pond bottom. But here, the entire duration is reduced by 2 months. The farmers in the area reported that it takes about 6 months for production, whereas the farmers in Dhaka, near Kotia, there is a village called Dhaka, where the makana business, a very sprawling business is there. So they are generally reducing the time. Entire span of makana cultivation in pond that is being reduced under the daily practice. Makana has a good quality of protein and starch and is almost fatless. It is a good source of micronutrients. It is used for curing a number of human ailments. Thus, it could be held as a significant nutraceous. It is almost fatless. 0.1% fat is there. So it is in high demand in affluent countries who generally do not allow the import of fatty food substances from other countries. So for affluent countries, makana is a good option. Those who do not want fatty products, for them it is a good option. Yes, I would like to show you makana-based integrated aquaculture. Just through science and technology, we have to find out number of animals. Just see. And till now, till about a decade back, there was a practice of capture fishing. Capture fishing means just go and take out from the system fishes like singi, mangu, kabai, laichi, etc. All these are black fishes and all these are air-breathing fishes. They come to the water surface, engulf air and go back to the bottom, which is quite rich in nutrients because nothing except seed is taken out of the system, and all the other parts they remain in the muck, in the bottom. So the bottom muck is very rich in nutrients, and that helps the air-breathing fishes survive in such a system. But now, since the option is on making Blue Revolution a success in our country, so all sorts of the value for raising fish production a success, a possibility. So under the makana system also, now the farmers, they are giving sufficient open space, either in the middle or on the side. This allows the carp fishes to derive oxygen from the soluble oxygen in the water. So they survive in that way. And it is again, I think old teachers, they are might be knowing Professor J.S. Kattamunshi, who was professor and head of geology department in Bhagalpur University. He was an FNA, fellow of Indian National Science Academy. And it is who discovered the process of air-breathing fishes, that is accessory breathing apparatus in these fishes. It was a monumental work at that time, and he got much recognition in India as well as abroad for his discovery. So it is these air-breathing fishes who are survive naturally, but today even carp fishes, they are being integrated with the system. So this is good exercise that is going on now. Let's see another big point. Big tank, the Jhanjharpur, the Kansar. Let's see how big the tank is. I will take a single photograph. But there are two photographs of the same tank. Let's see, open space is there. Yes, there is a chorus, air-breathing fishes, being integrated with carp fishes. Photograph taken from the case with the of the fishes and technically works. That is now bottom feeders. Yes, one thing, it is of evolutionary significance also. Makhana plant is of much evolutionary significance. Students of botany, teachers of botany who are here might be knowing about dicots and monocots during evolution. But here in Makhana, we have both the systems. We have both primary, these cells are coming out occasionally. But since it is a U dicot at the borderline of monocots and dicots, so here we find both the primary as well as tetrameric, 3 sepals as well as 4 sepals. Just see how it has flower coming out after maturity. It has 4 sepals. So both primary as well as tetrameric is found in the case of this one. Now here, this is the property of it can shout about 10 to 12% protein, but essential amino acid index is quite high, near 90. So it makes it just comparable with the amount of protein in fish and mutton. Which biological value is 55. It has all the good chemical scope. The number of other proteins are there, like not in a very good condition. Next, patents. A number of patents have been granted on Makhana in India as well as in other countries. So see, even today, bamboo appliances, which is called OGA, at least 3 persons are required to operate it. Bamboo appliances called OGA used for Makhana harvest. I think this condition is not there in your bed because here it is cultivated in field condition. But in non-condition, these bamboo appliances need to be operated. Just see, Naan is there. Naan is also used for collection of fish. It is used for collection of seeds as well from the pond. Just see, a vine used in the harvest appliance where the local values is. Now, plastic ropes are used. Makhana harvesters on migration. Yes, fertility is created seeds for seed powder and seed pods. We have aid over the seed of Makhana. See, that aid is very good, tasty product, and that causes the seeds to float for a few days on the water surface, and then it goes down. So the pods also they float on the water surface. They have to be stirred. Continuous study has to be done to then the powder and just go to the ground. This property has been exploited for making proper drug delivery of some other disease to require protection to the body. Tissue culture also has been very successful in this plant. Earlier, tissue culture was not successful in this Makhana plant. Professor H.Y. Mohan of Delhi University, he tried several times, but since it is an aquatic plant, there is much of infection in the tissue culture process. But it has been very successful. My one PhD student has done it successfully. Yes. Combination of dipole and monopole characters, a number of characters are there. A plant has 2 types of flowers. Makhana has got 2 types of flowers. One is histogamous that does not open, that does not come to the surface. And there is one exogamous flower. Just show how the exogamous flower of pollinator shows like this, and another exogamous flower shows like that on right part. So just like pollinator, Makhana also has got 2 types of flowers: exogamous that comes above the water, and histogamous that doesn't come above the water, and even does not open. Let's see, there is a saying, मिथिला में एक कहावत है कि मखाना के पत्ते से मुंह पोंछ के आई, मखाना के पात से मुंह पोंछ के आओ। एकर मतलब ई भेलै जे क्यों जे आदमी अपना के बड बोस्ट करे छे जे हम बड काबिल छी, तो ओकर दोस्त ओकरा कहै छे या हास्य प्रयास के जे संबंध छे ओ ओकरा कहै छे जे मखाना के पात से मुंह पोंछ ले आओ। मतलब मखान के पात में बहुत कांट रहे छे और वो कांट सीधे गड़ै छे, क्यों एकरा छुओ के साहस नहीं कर सकै छे। We cannot dare even touch the plant. है ने, तो अहाँ यदि करबै मुंह पोछबै तो सौंसे अहाँ के लहू लुहान है, खूने खूना में है, ई काज अहाँ नहीं कर सकै छे। तै ओकरा कहल जाय छे कि मखाना के पात से मुंह पोछ ले। कहावत एरिया में छै कि नहीं हेबे करतै, इहो मिथिले के पार्ट छे। that ladies who release initially seeds, ladies residents after floating for a few days on the water surface, lose color and thus facilitate the seeds to settle in the bottom. On the cube, you can see how spiny it is there. I have come to know that in China, spineless variety of banana plant has been developed, and it may take a few years if the result is to the point. It is correct. I think it won't take much time for this findings of research to come over to this place also, to our area also. And if a spineless variety of makana is developed, I think it will be much easier. Its cultivation will be much easier. Just like in the case of Singhara, Trapa, spineless variety of Trapa has been developed. Earlier it was four spine Singhara, 4 कांटे वाला Singhara होगा. उसके बाद 2 कांटे वाला Singhara आया, 2 horn, 2 Singhare वाला, and now spineless Singhara has been developed. Similar researches are going on how to throw on the surface after the releasing of fruit, just golden color, seeds floating on the surface. Seeds after developing of age chapta ho jata hai, and then it looks like this. The fruit of banana after removal of fruit, manually, the fruit has to remove manually, and then it looks like this. Entire placenta already the seeds, regulated seeds. Just see, ethnic significance of banana. Just be used for God carving sacred thread. There is no body. Banana star is considered, banana is considered sacred, and its solution is used for making the body. And in our marriage system, Allah is there, and you cannot think of process being organized without body. Just preparations for today, a number of preparations are being made from this component. One of us acts as a component of Havan Samagri. Havan Samagri, it is used in Havan also. Just being another item earlier, Havan also is a component of Havan Samagri. Then again, preparations are there. The expenditure incurred over cultivation is very modest, and the seeds left over after harvest that remain as a crop or a seed. The only expenditure is bringing out to overgrow, transforming into sparse area, insecticide, and collection of the first seed from the pond bed during harvest. The local fishermen, rear fish, control the great banana, and their livelihood is dependent to a large extent upon the combined cultivation. That's the case. Yes, the seed. Processing of seeds of the develop or poppy. Seeds are being read just by divisions and plants. Then they, after harvest, it is post-harvest process, and it is closed up. Right? Then seeds are being planted in sun, ready at the side of the seed. Then first roasting, then second roasting, then making of seed coat by coffee, food and melon, then ready of lava, then packaging and marketing of the lava, different like that. The seed, this is the case, and they can post-harvest production process. There, then this is the area of how they migrate, internally migrate from the Bangladesh to India, Katihar, and even to Saharsa. I think it is fishermen from the Bangladesh who come to this area for harvesting purposes. So, yes, like that. Intensive labor is required during home technique, and the temperature of the 90 is required. Seeded to Diana as defined as sheep or poppy, the modification of seeds. The new system facilitates continuous roasting with poppy. It consists of single layer and roaster. This is the river and poppy. It is the stage of the machine that has been made and seeded to Diana for making a pot. Good quality of banana, usually for Rasgulla or Lava, is used solely with 4% of pot. Otherwise, it is 5% of the medium quality flora and we don't get 2 meters. Such several items are prepared for this part. Wheat and sweet. The wheat, halwa, dal, makhani, kofta, aloo, etc. These are different items that are being prepared. A number of community to speak and make it say better type of business similarly business of now it is being exported also. Darbhanga has now an operational air force. Targus this is now ready, and Makhana is naturally exported from Darbhanga to other countries. Makhana is a macrophyte cultivated as a crop. It is selected by the body of crop, but its bioproperties are available in different parts of our India, China, Japan, Korea. Indeed, the celebration is alive, it is almost madness. It is a good sort of micronutrient. It is used for curing a number of human ailments. That's why it is called yes, nutraceutical formulations to be better. I think in India, these parts are prepared for pregnant women, lactating women. The Diffusion Technique has been prepared in China. The herbal supplementary has been prepared in Korea. All these preparations, they do. Yes, I already said on skills, the medical system, or coffee and deforestation of pharmacy, and de-allergenic property of the herbal provisions to treat airway infections. Any other crop, Makhana also invites. Rather it suffers from the attack of a number of diseases, including fungi and insects. Yes. शुरू करें? चल रहा है. A number of fungal and insect infections and associations are there in the case of Makhana crop. ये move करेगा?
Internet.
Just like any other crop, banana also is attacked, banana palm is also attacked by a number of pests, fungal pests, insect pests. So, and these two fungal pests are there. One is light, caused by material, and the other is mold. Yes. Branding major insect There is some problem. A number of entrepreneurs are coming in the field, and for them, storing makhana box for a longer period is problematic. They are looking for ways to store the makhana box for a longer time, and that should be non-toxic to the humans also. I have advised them to use vetiver oil for the purpose. That is vetiver oil is edible and it may provide some rescue. We have been under radio and applying to see how the damage is up there. We have been called to see how problem because are with the two problems. To see how the small balls are there. That's the real small ball and the right power showing the sport around. It's just a big case one ball. Just see this is total type of beauty. It is and a big drop, a big total type of beauty. This looks like this. Then we have larva to see how the market has been cut, how the market has been marked out. It was the case on the market of the university also showed networking spots caused by fungal life. To see how the case looks like. जैसे पत्ते को पोस्ट करके और उसको मैनेज करना जैसे दिखाई then a group of neuromuscular specialists and neuromuscular development how to investigate and apply to see the larva of neuroplasticity coming out of the case to see the types of neuromuscular development under portion that's how we are dealing and the portion of the trees that we are in that is associated with the flower of the plant. Posterior portion of the trees, right? Then the of the rest of our group is matched with the root, like root. Thus, the surface of the material where the trees showing a combination of astrophores and their legs. Astrophores are there in response to the vertical surface of the leaf. The strong cycle of the network, how it looks like year-round crop cycle is there. Then this is the water, let's see, storage. This storage, how the set is there, then it is the of housing, attack by storage and see all of and ammonia and one new center next to the Zeta and a new center for this the entire process of we explain let's see how they are creating fish. See variety of two variety one short variety there are some and we develop by two centers. Now we are doing them that we have another growth of peak as I said after peak slump. And we graduated from another time. Just see, it will be 10 months of a look like January. Just see, this is the case, and you will not be unable to see the right side how the farmers they are collecting fruits from the people takes. The coffee is not there, artisan is not there, they just collect fruits. And in that all these sides, they are from people. Just see all the fruits are being sold in the Imam market, run by women. They are selling fruits. It is being what geographical indicator tagging under the name of we have to say how this art is used and there is a good market for just see after Prime Minister about Karnataka district and that ODOP, one district, one product. It's a big from Prime Minister. So he also organized a meeting with state team members. So he has a season to team members on receiving PA award under one district, one product scheme. Now I would like to bring to notice that your Saharsa district also. Saharsa, Supaul, and Aranya and Kandhar. These are 6 districts. 6 districts have been earmarked for ODOP scheme. So, government is providing all possible support to the growers, makana entrepreneurs in 6 districts. So, just see the flower, how the flower looks like, and the garlands. Makhana garlands also have good prospect. Dr. Vinod Kumar Gupta, the previous head of he was talking about how the Makhana garlands would be projected for a use wedding. ये जो बरमाला का जो कार्ड होता है उसको पूरे देश में प्रचारित करने की आवश्यकता है। अभी हम माला या और माला का प्रयोग करते हैं, लेकिन टाइम ऑफ वेडिंग हमारे माला है यदि हम इसकी मार्केटिंग ठीक से करें तो यह भी एक मार्केटिंग का इसका एक ऑप्शन है। So with this, I would like to thank you for inviting me to this talk. कुछ पूछना है कि कहीं माइक वहां आइए आइए
Dr. Zeta, I will be assessed for its support and seminar, and I will request you for the long time to our work in Makhana. I know so I am very impressed with you very long time.
I am highly interested. First of all, I am highly thankful to principal sir, MLT college sir, and all the members of the organizing committee to provide me such a great opportunity to speak as a resource person. I believe that I am the last speaker of this seminar, and I have allotted around 30 minutes time to speak. And I will try my best not to make you bored in this session. And in next 30 minutes, I will try my best to make you all conscious about my lecture. So, my lecture basically I am a synthetic organic chemist. I synthesize small organic molecule and then I go for the application of that. In which field they can be applied? Maybe they can be applied in the field of drug and medicine. Particularly, my last 15 years training is in medicinal chemistry. I synthesize molecule for several targets, and among them one is the diabetes. For diabetes, there is one molecule, Ebola. That is the first molecule which was discovered in India and marketed in India for diabetes. That molecule was found. So, in today's session, I will explain the synthesis of phosphorus dendrimers. Actually, dendrimers are very fascinating molecule. For a chemistry person, they will understand more that these are big molecules, but not polymers. These molecules, but are synthesized in step by step manner. This gives a symmetrical and beautiful structure of molecule. Once you will see the structure of molecule, you will realize that what is the driving force behind this synthesis of this molecule. See this big molecule, how symmetrical it is. These branches just double 1, 2, 4, then at each step they double. So these molecules have very fascinating symmetrical structure, and this gives some motivating force to synthesize. This is one reason, but these molecules have too many applications. Like we have to play with the surface. What is the surface? On surface, if I make it cation, if I make it anion, if I attach some drug molecule, some polar molecule, some bioconjugate, the application is drastically changed. They can be used in electrodes, they can use anti-cancer agent, nano device, DNA chips, anti-prion, drug delivery, medical chemistry, so many applications. That's why this molecule is fascinating. Just by synthesis, this molecule we can enter in anything. And particularly this derivative is phosphorus based. You see, here is the tool we have phosphorus. Phosphorus, as a chemist, we know it has three valencies, two types of valencies, 3 and 5. 5 is more stable. Working with phosphorus is a little bit tricky and difficult. Why? Because plus 3 is not stable. Easily working it will oxidize to plus 5. That's why the synthesis is very tricky for this derivative. So, as I explained, by changing the surface, if I have an ionic surface, this shows anti-inflammatory. Neutral surface, it has fluorescent property. But cationic surface, application is changed. It works as a DNA RNA delivery system. So this slide may be very useful for students, PhD students. What is the principle of dendrimer? We have a core, a central core with nitrogen, phosphorus, what we have seen. Then how step by step manner it will go, you see. Then first we need to activate. After activation, we get coupling of just same core again 3 times. So this will generate the generation 1. You see here 1, 2, 3, but here 1, 2, 3, 4, 5, 6. And now We can double it again. You see, I think then double it. You see, now 12. And so on. We can keep on doubling that and getting the basic structure. This is the challenge of synthesis. This is the very important reaction before starting the synthesis of phosphorus and Ppi. This is monomethyl hydrazine. Hydrazine is very common chemical in aeroplane fuels. And this is hydrophosphate chloride. Phosphorus is +5 state. Is a sulfur. And this reaction is -60. Just let me share you my experience. When this reaction was at -60 means -60, not -59, not -61. We did not get the product. We have to maintain strictly -60. This is a very tricky and difficult reaction. The first one. Rest one we can manage, but this is very tricky. Because at -64, chloroform will freeze, and at -59, we will get the impurity polypeptide. So see this slide, how pentamer is growing. We have a core and we have 6 chlorine atoms here. Just in the first step, I attach benzaldehyde and 6 benzaldehyde on the surface. Then again hydrazine attached on the surface. Then again attach the benzaldehyde. Then again hydrazine on the surface. Then again benzaldehyde. How the system is going? In this way we can build the dendritic core. Since we have a phosphorus, we analyze by phosphorus atom. In phosphorus atom, we can see the peaks of phosphorus for generation 1 dendritic. We have 2 peaks. For generation 2, we have 3 peaks. For generation 4, we have 4 peaks. Because at each level, phosphorus is getting added. This is the work when we were doing in laboratory, and this is a very beautiful white color powder. We can just prepare and crystallize in hexane. We can filter it. It is very stable, no hygroscopy and no air sensitive. We can keep it for years. So now, why do we prepare this big structure? So much effort. Why? Because now we have 3 places to work. One is the core. This is the generation 1, 0. This is the generation 1. We can change the chemistry at the generation 1. We can change 0. We can change the core. This is the beauty. By changing this chemistry, we will change the application of this enzyme. That's why these kind of enzymes are patentable, and they have already patented in several other kinds of applications. One of these enzymes is in phase 3 clinical trial for osteoporosis. Maybe very soon it will cross. So this what I showed you. So how the synthesis is started? We get the first generation enzyme, and to make the surface cation, this time we apply the purity amine hydrochloride, and we cover the surface with the amine, then make a hydrochloride salt. Hydrochloride salt is very crucial because before hydrochloride salt, it is not soluble in water. हमको कोई ड्रग बनानी है, दवा बनानी है, the most crucial property is the solubility. Just for example, curcumin. Curcumin is an active constituent of turmeric haldi, and everyone knows. Curcumin के साथ problem क्या है? When we take जब haldi लेते हैं, it is not solubilized in water. So body में वो absorb नहीं होती. उसको to make the drug-like properties, we have to make a water soluble. So by making this hydrochloride at the surface, now this gives the water soluble. Once we will take, it will absorb very quickly. That affects the bioavailability and efficacy of the molecule. So in this way, we were able to prepare several derivatives of dendrimer. And these kind of surfaces we have differentiated. In one, there was a piperidine, pyrrolidine, propylene, and several other types of amine varieties to make a hydrochloride salt. See, this is the structure of first generation dendrimer, which was patented for tuberculosis. This dendrimer molecule was having very good activity against tuberculosis. Tuberculosis की problem क्या है? The problem is the last molecule which was developed for tuberculosis. It was 1964. 1964 के बाद कोई दवा नहीं बनी। वाह, क्या हम लोग काम नहीं कर रहे हैं? या no one is working for tuberculosis? No, everyone is. But the problem is different. It is the microbacterium. The microbacterium tuberculosis is very different. Identifies the environment, the molecule required, and generate the resistance. And once it generates resistance, now we need some other kind of molecule. एक बार मारने गया, उसको उसने पहचान लिया कि मारने आया, अब हमको कोई दूसरा सही देखना पड़ेगा. तो for tuberculosis we need new molecule rapidly at a fast stage. So this was a different molecule, but it showed very good activity against diabetes, and presently it is under screening in anyone. This is the same kind of dendrimers, but they are second generation dendrimer. The earlier slide was first generation. The molecular weight increases. This is zero generation dendrimer. See, the size is a little bit small than the previous one. We have only 6 pyridine groups on the surface, and it was surprised to us that the small size dendrimer was more active than the big size dendrimer. So this is the synthesis of anionic dendrimer. Okay, the previous one was cation. On the surface, we were having positive charge, the cation, and this time on the surface, I am using the minus charge, the anion. And let's see what kind of change in the activity by changing the charge on the surface by cation to anion. The synthesis is a little bit different from the earlier one. Like this, 4-hydroxybenzoate is reacted with chloroamino, then this undergo under galactic fields reaction to make the organophosphates. Organophosphates hydrolyze to make the acids. Now we get this monomer. So what we will do? There is a phenol liquid, and this phenol will be grasped on the surface of dendrimer. See now, synthesis of dendrimer. We have dendrimer and we have chlorine atom. This phenol will substitute on the surface to get the anionic dendrimer. So now we get the organophosphates on the surface, and these phosphates can be hydrolyzed. Now see, we have acid, not phosphate. We have phosphoric acid, and this acid will make anion. Once it will produce, and this particular molecule is in phase 3 clinical trial or osteoporosis. So see the difference. The previous one, which was cation, that was for tuberculosis, and when it is anionic, it is again for osteoporosis. So by changing the surface, the application is changing. This is some of the biological result how the cultures of tuberculosis. It is showing the inhibition of mycobacterium. This paper recently has been published in American Chemical Society, and this slide is just showing by increasing the size of dendrimer. We are going with a decreasing size. Generation 0, 1, 2, 3, 4. Once we are decreasing the generation, we are going to bigger to smaller, the activity is increasing. Here the activity is more for generation 0, activity is less for generation 1, then again less for 2, 3, 4, it is constant. So size of dendrimer is also affecting the property of dendrimer. So I will conclude very shortly. So what in this work I have shown that we have prepared the cationic dendrimers, anionic dendrimers, and amphiphilic dendrons. Dendrons I have not shown here, time constraint, but dendrons was having other application like gram-positive negative inhibition bacteria. But for cationic it was antimicrobacterial. For anionic it was not antimicrobacterial, it was osteoporotic. This here I will conclude my talk. This was a team where I was working. I am highly thankful to my collaborators, Professor Jampia. He is a very old guy like him here, but very dedicated professor. I learned a lot from him. At the age of 88 years, very active. And this engineering cabinet, he was working with both of them. Professor Saraswati was from the University of Paris. Kishore Srivastava sir was from Srinagar. He was performing all the biological activity regarding tuberculosis. I am thankful for all the funding agencies for this product, the Cervicula, CNRS, University of Australia. I am highly thankful to my present university, LNW, where I am working, and due to that only I am able to present my lecture. I am very thankful to you all for patience hearing, and if you have any question, I will try to answer. Thank you very much.
Thank you for your advice and your guys on the dendrimers. We discussed about the cationic dendrimers, anionic dendrimers, and amphiphilic dendrons. Sir, I have a query. Is the possibility of beta in the case of dendrimers, the dendrimers molecule having both the cationic and anionic molecule in the same molecule?
In the same molecule, it can be. See, since characterization is very challenging part in dendrimers. In few of the molecules, we have even 1,000 carbons. So if I have 1,000 carbons and in dendrimer, we are assigning all the carbons the PEC carbon amount. So we can build the backbone and the skeleton. Now, if we talk for the surface, if we have a charge, then how to confirm that we have charge? This is your question, and how to differentiate the cation and anion. So, for confirming the charge, the first clue we get by the solubility. Just we add the hydrochloride in stoichiometric amount, and once it is protonated, it dissolved in water. This is the first clue that we get the hydrochloride salt. Now, how to confirm? That is by measuring the zeta potential value. So once we have a charge on the surface, it is a charged particle and it will have some potential under electric field. So by that potential value, we can assign the amount of charge and the cationic or anionic. Thank you, sir.
Any audience, any question from audience?
थैंक यू सर आपने रुचि का जानकारी को दे दिए हैं इसलिए मैं आपको धन्यवाद देता हूं और मैं कहूंगा कि ट्यूबरकुलोसिस के सबसे बड़ा प्रॉब्लम है कि इसका लक्षण बहुत खराब है जब तक ये स्प्रे करने के शरीर में और कर सकता है तब डॉक्टर कर सकता है ये कहना है कि आपका ये जो रिसेंट स्टोरी है क्या उसी लक्षण को डिटरमाइन करेगा पहले और दूसरा क्वेश्चन ये है कि आपने फॉस्फोरस को सिलेक्ट ऐसा क्यों दूसरी बात है कि ये एबीपी मॉलिक्यूल्स है और ये मॉलिक्यूल्स में काम क्या कोई कोरिलेशन है क्या इसमें? थैंक यू।
First, I made it very clear that to cure a disease, अगर हम किसी disease को cure कर रहे हैं, this is not a single hand part. ये single hand का part नहीं. Like if we talk about the target of disease, like in tuberculosis, like in targeting microbacteria. ट्यूबरकुलोसिस की दवा जो डॉक्टर दे रहा है वो ट्यूबरकुलोसिस की पैथोजेनेसिस के बारे में जानता है, ट्रीटमेंट लाइन के बारे में जानता है, लेकिन जो ट्रीटमेंट की दवा पे काम कर रहा है he knows only microbacteria. Suppose I am a medicinal chemist, so my target is mycobacterium, not the person who is infected with mycobacteria. So for appearance of symptoms is not my objective. My objective is to how can I kill or restrict the mycobacteria. Second one is for phosphorus. Very interesting story about this guy. Actually, the place where I was doing this work, the place is Toulouse. Toulouse is a small city in south of France, and it is very famous for Airbus headquarters. Airbus is a company who manufactures Boeing and Airbus. दो कंपनी एरोप्लेन बनाती हैं, उसमें से एक वो टूलूज में हेडक्वार्टर. Due to presence of Airbus company, we were having the availability of this chemical. I will say. In the very first slide which I was showing, this monomethyl hydrazine. This monomethyl hydrazine is a banned chemical. Due to this explosive अगर इसको बम बनाने में या अदर एक्सप्लोसिव चीज में इसको यूज कर सकते हैं, इसलिए मोस्ट ऑफ द कंट्रीज में ये बैन है. उस सिटी में ये अवेलेबल इसीलिए था कि वो एरोप्लेन फ्यूल से यूज होता. तो इस डेल्टिमल की केमिस्ट्री है, द केमिस्ट्री ऑफ डेल्टिमल इट स्टार्ट विद द स्टोरी ऑफ दिस मोनोmethyl hydrazine. Now this monomethyl hydrazine, how it can be grafted or how can be utilized to form the core. The reason is the this Cl bond. कोई भी रेजिन बनाने के लिए हमको एक ऐसी केमिकल मॉलिक्यूल चाहिए जो बहुत रोबस्टली हो और बहुत सेलेक्टिव हो. अगर हम कोई भी रिएक्शन कर रहे हैं तो और कोई फंक्शनल ग्रुप को अफेक्ट ना करे. It should not affect any other functional group and directly it should go on the Cl bond. This Cl bond strength is very appropriate for the grafting of phenol to the phosphorus. If I will change the system other than the phosphorus like carbon, then this bond is not very selective. And in the dendritic synthesis, it is very challenging to control the reaction. हमको reaction क्योंकि जीरो स्थिति तो दो बॉन्ड है, तीन बॉन्ड, लेकिन जब हम चार में जाते हैं तो 25 जगह होती है जहां रिएक्शन हो रही है. तो हमको ऐसी साइड चाहिए जो क्वांटिटेटिव भी हो, सेलेक्टिव भी हो. मतलब सारे 25 रिएक्शन हो जाए और कहीं रिएक्शन ना हो. दिस इज वेरी सेलेक्टिव. इसको खोजना कि ऐसा कौन सा बॉन्ड है और ऐसी कौन सी साइड है, दिस इज द मैटर ऑफ वेरी हार्ड एकेडमिक रीडिंग. इसको बहुत सोच के केमिस्ट्री डेवलप करना पड़ता है. In fact, there is only one group I can say by very sure in the world which is performing this reaction, and that group is the same laboratory. This is the reason why there is no competition on this reaction. No
group is working. If someone wants to work, he has to contact this group and ask for a technician. Okay, come into my university and show me how to do this reaction. That's it.
Obviously, sir, it is very hard to control these reactions. Or you are saying that there are over 25 sites, reactive sites. The molecule will go there. To control the site is very hard. The one thing I want to know, the network there are many ones. Basically, there will be many hydrogens there. And maybe actually amine substituted. So hydrogen on the amine group also. We are very curious to know the spectral report of these radicals, few radicals.
Yes, I have. I have. Just this publication just published in Biomacromolecules. I will show you the paper and spectral details. Visibility is not very clear, but in this picture you see, this is the same scheme which I was showing for cation antennas. Starting on 2025. Now I will open the spectral part. This is the solar proton NMR for first generation rendering. You see here you have the filtering piece, the 6 numbered aliphatic cyclic amine, and here these are the aromatic protons of the enthalpy band. This was further characterized by this is depth experiment. DEPT 135. In this experiment, we get the quaternary carbon and secondary carbon at the bottom. CH2 are here and all the CH3 are here. Quaternary carbon will disappear. And again, this is the carbon NMR C13. This is the HMDC. Hetero multiple bond correlation. This is a two-dimensional NMR spectroscopy, and this was used to assign the exact conformation of the ring at the surface. In which carbon is interacting with which carbon. This is the HMQC. HMQC and HMBC are a little bit different in quarter pole movement. This is again the 6 number ring NMR. I will send you all the supporting information if you want. Thank you. If anyone has.
Thanks, sir. Anybody from audience, participant, teachers? Okay. Okay, thank you, sir. Thank you, Dr. Tripathi, for your nice talk. And one information I want to share with the participant, especially for the chemist, if you have to want to work on the animals, you don't have to go to
Good afternoon, everyone. It has been such a great honor for us to be a part of the wonderful event on behalf of our organization. As chairperson, I would like to extend my heartfelt gratitude to our esteemed guest. A special thanks to our distinguished speaker for being here with us today. Respected Speaker Dr. B.D. Tripathi, Department of Chemistry, LNM Darbhanga, has stated in his lecture that inadequate bioavailability of the drug is the main factor for failure of treatment of tuberculosis, which leads to the development of a new antimicrobial drug that should be minimize the treatment of the time period. He explained the development of monocytotoxic polycationic phosphorus binders, generation 0 to 1, with pyridinium group on the surface of the anti-tuberculosis agent. This topic was very relevant today due to its own value. I extend to my great thanks to him for this valuable speech. Our next speaker was Dr. Vidya, who explained here about the makhana, which is the basis on which base the sustainable development of Bihar is going on. He explained how makhana is useful for health. It contains various nutrients such as starch, proteins, etc. It is rich in phenolics, flavonoids, minerals, and antioxidants. Northeast part of the India, especially Mithila region, is a great source of makhana. It is due to its geometrical conditions and geographical conditions which leads here, like waterlogging is the main factor for the makhana development. Besides Mithila region, its fossils are also found in other part of the country. Not only this country, it also obtained outside the country like China, Korea, Japan, etc. Our central government and state government has made plan to developing of growing the makhana in the Mithila region to make the Bihar as a brand. His lecture was very excellent, and I extend my gratitude to Dr. Jha for being with us. Finally, sincere thanks to all participants present here and all head of the departments present here. And finally, I would like to thank all of you very much.
Thank you. Dr. Krishna Sen. He summarized in brief what the earlier university was told the facts before us. Now I would like to request all the respected persons sitting in this hall to move towards the lunch or safety. अगर मैं कहूँ लंच की तरफ चले और मैं अपनी तरफ से इन दोनों वक्ताओं को बहुत बहुत धन्यवाद देता हूँ कि देखने के लिए प्रिविलेज करते हैं। थैंक यू।
लंच के बाद और प्रेजेंटेशन आएगा, जो पार्टिसिपेंट हैं उनका और प्रेजेंटेशन होगा, आफ्टर और प्रेजेंटेशन थैंक यू।
बदले से तो छै कहाँ हाँ? ऑफ क्या था
प्रैक्टिस कर लेते हैं एक बार यहाँ।
हाँ हाँ अच्छा कीजिए।
माहिन आप प्रैक्टिस कर लो एक बार। सही बोल रहे
हो गया।
अरे वो पढ़ लेते हैं। यहाँ पढ़ने के बाद कैसा आता कुछ वो करता है, यही देख रहे हैं। What is the most precious gift of nature which is essential for the sustaining all form of life in India? There has been a tremendous increase in the demand due to ठीक है चलो अंदाजा लेने दो।
हो गया। Headphone cut.
Yeah. यहाँ देखिए दिए पूरे बना लीजिएगा नहीं
Hello. Welcome. Monji. Yeah, okay. हो गया आज तुम आ जाओ आना हम फंसते अपने लोग हैं वो आंटी ये ऊपर लिखा हुआ साइड लिखा हुआ
वेलकम से भाग ले हैं नामो नहीं गिरा छे हां वेलकम क्या दियो हेलो Okay, next we are going to start our technical session where oral presentation of participants we are going to give. So beginning with OP 31, we have Hemant Kumar Das. Are you there? Mr. Hemant Kumar Das, Anil Kumar, then Dr. Raju Kulkarni, okay. From the participant side, I think Aishwarya Shekhar is online. So are you ready to give your presentation? Aishwarya Shekhar? And Ashwini Kumar? Babli Kumari, are you ready for the presentation? Sachi Kumari, are you ready for the presentation? एकरा अनम्यूट करियो के ऐश्वर्या से वो मैसेज करके जो हम म्यूट करा दे छी पुट इन वेटिंग has to start with मैसेज की है ना
Yeah, good afternoon, sir.
Yes, you yes, Aishwarya Shekhar, you can start your presentation. Your time limit
Okay,
is
sir.
only 3 minutes.
Sir, I have to share the PPT also. Hello?
Hello? Yeah.
Yes, sir, I have to share the PPT also.
Yes, yes. Please share your PPT and try to complete within 3 minutes.
Sir, host disabled participant from screen sharing. This message is coming from my side. Sir, again, the same message is coming. Host disabled participant screen sharing.
You start your presentation.
Okay, sir. Good afternoon to all of you. Today I am here to present on the topic of future and prospects of digital media in the context of green cloud computing and IoT devices. I am working as a research scholar in Galgotia University, and I am doing work on IoT. But here it's related to digitalization. So digitalization is defined as a process of converting information into the digital format so that it can be used on computers. In the past decade, we have seen massive digitalization. All our information, which were previously stored in the files and notebooks, are now available in digital format, which we can instantly access it from any of our devices. People can use their phones, tablet, and laptops to do the things like grocery shopping and banking, etc. The total time spent on these electronic devices has risen dramatically in recent years as all the work and social lives are moving online. For transferring everything online, cloud computing is being used. As we know that cloud computing is a type of computing that relies on sharing computing resources rather than having local servers. Cloud is a very good initiative taken by those industries which deals with the data sharing or saving. But excessive use of cloud computing creates emission of harmful gases that gives a very bad impact on our environment. So for implementing digitalization properly, we must have to move from cloud computing to green cloud computing. So green cloud computing is a term that use the resources and the equipment that are recycled. It is very important to implement green cloud computing everywhere to reduce the emission of harmful gases like carbon dioxide and to lessen the heat produced by the servers and hardware used in the cloud computing for storing the data. I am working on Internet of Things, and it is also related with digitalization. So Internet of Things was coined by Kevin Ashton in 1999. IoT devices are those devices which are connected to the internet and is used for transferring data or information from one place to another using the internet. The extension of internet connectivity into physical devices, IoT is a world where all the smart things are connected and can communicate with each other. IoT device is the future of digital India. Prediction that by the end of 2025, 1 people will be connected with minimum 5 IoT devices. Hello, my voice is coming.
Yes, you are audible. Please continue.
Okay. And then what is digital transaction? Digital transaction nowadays is the result of digital India. Digitization is the finest way to move in the direction of cashless economy. Doing cashless transaction in India is an effort to move from traditional way to modern way by implementing the digitalization in India. There are many reasons for the people choice to do digital transaction like convenience, trusted digital technology, and all these things is possible because of the initiative taken by the government to implement green cloud computing and IoT devices everywhere in India. India is planning to introduce digital currency, and it is announced in the current year budget also by our finance minister. And it can be made possible only with the help of IoT devices and the green cloud computing. So we have to focus on the green cloud computing and IoT devices to implement digitalization in India on a good pace. It's because of the Digital India initiative. During the COVID-19 lockdown, we are able to attend our lectures and classes on Zoom and Google Meet, and it was only possible because of this cloud computing, internet, and IoT devices. Thank you so much. Any query from your side? Sir, my presentation is over from my side.
Hello, please continue. You are audible.
Sir, my presentation is over.
Okay, thank you.
Thank you, sir.
Okay, next participant, Aswini Kumar, are you ready? Aswini Kumar?
Yeah, I'm quite ready, sir.
Okay, please continue.
Honorable our Chief Guest, Chairperson, Professor Dr. A.K. Jha, sir, Professor Dr. B.K. Singh, sir, and Repertoire Dr. V.D. Tripathi, sir, Convener Dr. Sanjeev Kumar, sir, and respected all faculty members of this institution, who are they presented here with more valuable time among us. A warm good afternoon to you all. I am very fortunate that I got the golden opportunity to participate in oral presentation along with my abstract in International Conference on Emerging Trends in Science and Technology 2022 held in this organization. So let us what I have performed in our laboratory, that abstract is bioactivity of some heterocyclic compounds. So I am sharing my PPT. Synthesis and fungicidal activities of some 1,3,4 oxidazole or 3,2-D 1,3,4 thiazine. Abstract, we have taken 2,5-diaryl 1,3,4 oxidazole 3,2-D 1,3,4 thiazine 4 have been conventionally prepared from 5-aryl 1,3,4 oxidazole 2-aryl methyl and aryl dithiocarbamate compounds, and that's which are synthesized by general method from 5-aryl 2-chloromethyl 1,3,4 oxidazole. That's the first compound with ammonium nitrogen aryl dithiocarbamate of the second in presence of anhydrous sodium acetate. And all the title compounds have been tested in vitro for their antifungal activity against 2 fungal species, Collectoricum and falcatum and Fusarium all. Introduction. So first of all, I would like to introduce that present large number of research interests in synthesis of bioactive molecules, especially heterocyclic compounds. The heterocycles have a large range of applications as they used as agrochemicals. The annual loss of edible crops due to fungal disease in India found to be several million tons. We know that so many crop diseases caused by fungi, bacteria, virus, which can affect on the biological factors of plants, causing morphological and biochemical changes, which have been collected in the sculpture. The chemical used to control pests are termed as pesticide like fungicides, insecticides, herbicides, etc. Therefore, the development of novel and promising antifungal agents is still an urgent task for me, sir. In terms of biological potency, heterocycles have a specialized reactivity. The diothermocarbamates like Meneb and Zineb, which are the most important commercial fungicides, as we know. Rhodanine incorporating dithiocarbamate moiety is highly toxic to microorganisms, and they have evoked considerable actions. Similarly, it has been observed that 1,3,4 oxidazole nucleus is associated with a broad spectrum of biological activity like bactericidal, fungicidal, herbicidal, and insecticidal. Therefore, sir, it was anticipated that the combination of the two moieties 1,3,4 oxidazole first of the compound and dithiocarbamate 2 and produce title compound third with enhanced fungicidal and other biological activity. Now, in the next, materials and methods what we have been used. In this project, the title compound for it will synthesize by refluxing 5-aryl 1,3,4 oxidazole 2-aryl methyl N-aryl dithiocarbamate 3 with thionyl chloride. The compound third will synthesize by refluxing 5-aryl 2-chloromethyl 1,3,4 oxidazole, that is the first, and ammonium, what I have been used in the scheme. In the following reactions, I have been used. And fungicidal scanning, I would like to look at it. The fungicidal activities were evaluated against Collectoricum, falcatum, and Fusarium. Oxysporum by usual agar plate technique at the 1,110 ppm concentration using the M45, a commercial fungicide, as a standard. The number of replications in each case was 3 after 96 hours. The diameter of fungal growth zone was measured. The results were expressed in terms of percentage growth inhibition. By comparing with growth on control, the percentage inhibition is given by what I've been used the formula C minus T into 100 by C. That C is representing the diameter in mm of the fungal colony in control plate and T diameter in mm of the fungal colony in treated plate. Experimental knowledge. So melting points were determined in open capillaries and uncorrected. IR spectra in potassium bromide were recorded on a Perkin-Elmer 881 infrared spectrophotometer. Maximum per centimeter and proton NMR spectra and dimethyl sulfoxide D6 were recorded on a Varian EM360L 200 megahertz spectrometer using TMS dimethyl sulfide as internal references. And the 5-aryl 2-chloromethyl 1,3,4-oxidazole 1, what I've been used in the scheme 1. These compounds are prepared by heating chloroacetyl chloride 0.1 mole and derivative of benzoic acid hydrazide 0.1 mole on oil bath and the melting point of acid hydrazide until the evolution of HCl has been ceased. Ammonium nitrogen aryl dithiocarbamate, which is mentioned in the second of the compound, that had been prepared by the reaction of carbon disulfide 0.25 mole into the mixture of substituted aniline 0.2 mole and ammonium solution in ice bath. 5-aryl 1,3,4-oxidazole 2-ylmethylene aryl dithiocarbamate, third of compound. These compounds have been prepared by refluxing mixture of 1, that is the first compound of 0.08 mole taking, second compound 0.08 mole taking, and absolute 1 degree of alcohol for 2 hours. The desired product, this precipitated, was filtered out and washed this water and crystallized from ethanol, and as so on. Results and discussions. The fungicidal data indicates that all tested compounds showed a strong and moderate activities. It is interesting to mention from antifungal data that all the tested compounds for H displayed significant fungicidal activity at 1000 ppm against both the test fungi Calcodium falcotum and Fusarium oxyporum. But their activity decreases at lower concentration, that is 100 ppm and 10 ppm. It is most important to mention the presence of more electronegative toxophorus.
Conclude with it.
Yes, sir. Pardon. Is there any question? The title compound 4 also contain carbon disulfide group responsible for enhanced activity. This is in conformity with earlier observation that carbonyl and carbon disulfide group reduces fungi toxicity. In fact, the combination of some method bioactive nitrogen heterocycle like oxidazole and thiazine have been given more potent compound. And although dithiocarbamates 3H have a pre-formed open chain skeleton of 1,3,4 diazine ring, these were considerably less toxic than other cyclized product for H, where chain is closed, resulting in more compact and planar system. And thank you. I would like to stop my presentation here. Thank you all for your kind consideration. I have taken some references. I've been also mentioned in the PP.
Soni ji, please try to conclude within 2 minutes. Okay.
Yes, sir. I have concluded my presentation. Yes, sir. Okay. I have concluded my presentation. Are you audible my voice, sir? Thank you, sir. Thank you for your
Babli Kumari, are you ready for your presentation? Babli, are you ready for your presentation? Babli, no. Then Mr. Rana, are you ready for your presentation? Rana, are you ready for your presentation? Okay. Rishi Raj Ojha, are you ready for your presentation? Rishi Raj Ojha, are you ready for your presentation? Okay. Next, Sana Samsad. Sana Samsad, can you give your presentation now? Sana Samsad, can you give your presentation now? Sonam. Sonam. Sonam, are you ready for your presentation? Yes. Yes, Mr. Rana, you can continue. Rana, you can continue for your presentation.
Hello? Hello, am I audible?
Yes, you are audible. You can share your screen.
Sir, I can't share my screen. Host disabled participant screen sharing. I'm unable to share my screen. Okay. Hello. Hello.
Yes.
Good afternoon, everyone. My name is Dana Adam. Hello. Hello. Am I audible?
Yes, you are audible. Yes, audible to all.
I'm Dana Adam, District of Scholar, Department of Botany, Bihar University. Today I am going to give presentation on the potential impact of climate change on air pollution and related human health effects. Air pollution. Air pollution is basically the presence of foreign substances in air in excessive concentration which adversely affect the well-being of the individual or causes damage to property. According to WHO, air pollution is defined as substances put into air by activity of mankind into concentration sufficient to cause harmful effects to specific health, vegetation, property, or interfere with the enjoyment of its property. There are different sources of air pollution, both natural and man-made sources. Natural sources include natural contaminants, fungi, spores, ticks, bacteria, methane, dust, etc. Carbon monoxide from methane gases, volcanic eruption, forest fire, electric storms, and solar flares, salt spills from oceans, dust storms are natural sources. That is, man-made sources are emission from vehicles, rapid industrialization, agricultural activities, warfare, automobile revolution, deforestation, and burning of fossil fuels are the man-made sources. Air pollution and climate change are two sides of the same coin. Air pollution and greenhouse gases have common sources, although they may seem to be two very different issues. Climate change and air pollution are closely interlinked. So by reducing air pollution, we also protect the climate. Climate change may have an impact on local air quality. In many areas, climate change related atmospheric warming has the potential to increase ground level ozone, posing a future challenge for ozone compliance. Climate change's impact on other air pollutants such as particulate matter is less certain, although research is underway to address these unknowns. Pollutant emissions can cause climate change. Different components of particulate matter can either warm or cool the environment, while ozone in the atmosphere heats it. Black carbon, a particle pollution produced by burning, contributes to global warming, whereas the particulate sulfate cools the planet's atmosphere. Weather and climate plays a key role in determining air quality patterns over many times and space scales. Varying climatic conditions and meteorological variations have an impact on air pollution emission, transportation, dilution, chemical alteration, and eventual deposition. Climate change could affect the dispersion of primary pollutants, particularly particulate matter, as well as the development of secondary pollutants like near surface ozone. Climate change alone will increase mid-summer surface ozone in polluted places by 1 to 10 ppb in the future decades, according to combined GCM-CTM research. With the greatest effects in metropolitan areas, climate change has a more complicated and ambiguous impact on particulate matter than ozone. Climate change is expected to increase particulate matter concentration in contaminated areas by 0.1 to 1 gram meter cube over the next few decades, according to GCM-CTM research. Climate change-fueled wildfires may become a major source of particulate matter. Air pollution has a severe impact on human health, causing up to 7 million premature deaths per year. Ground-level ozone can exacerbate chronic respiratory illness and reduce lung function temporarily. Particulate matter exposure can exacerbate chronic respiratory and cardiovascular disorders. The integrated atmospheric and health modeling was used to study the possible health effects of ambient ozone and PM 2.5 over the United States. PM 2.5 induced premature death is around 15 times higher than ozone induced premature mortality, according to the findings. Basic atmospheric science research atmospheric science research on the relationship between weather and air pollutant is needed, as is improving air pollution models and their links to climate change scenarios, and closing gaps in understanding of exposure patterns and health effects. More than 100 million people in the United States live in areas where air pollution levels exceed health-based regulations. Climate change will aggravate present air pollution levels unless countermeasures to improve air quality are implemented. This increased air pollution would raise the risk of negative respiratory and cardiovascular health impacts as well as early mortality. Increased air pollution would also have additional environmental repercussions such as diminished visibility and crop forest damage. Thank you. Hello.
Okay.
Thank you.
Okay. Thank you. Next participant is Rishiraj Ojha. Are you ready? Rishiraj? Rishiraj, are you ready? Okay. Then Praveen Kumar. Praveen Kumar? Then Pooja Mandal, Raju Ranjit Raj, okay, anybody who are online and wish to participate, wish to give his or her presentation, can raise your hand or you can message me.
Hello. Hello. Hello.
Yes, Mr. Anil,
Hello.
you can give presentation. Yes, Mr.
Yes,
Anil,
sir,
you
I'm
can give
speaking. Sir, yes, sir. Sir, I just sharing our screen. Sir, how to share a screen, please? I try to facing some problem due to network, sir. Sir, just I try to sharing our screen, sir. Please, please, sir. Please, sir. Please, sir. Sorry, sir. Sorry. Yes. Yes. Sir.
Okay, Mr. Anil Kumar Pandey, please continue. You are audible.
Yes, sir. Yes, yes, sir. Yes, sir.
So you can continue now because we have limited time.
I am audible, sir. I am audible.
Yes, you are audible. If you are unable to share your slides, then you continue your oral presentation.
Okay, sir. I give you give permission for oral presentation only, sir. Due to network problem, I unable to facing on screen sharing. Okay, sir. Sir, respected sir and all present here, that is very opportunity to present our abstract among this. That first all good afternoon to all of you in this international conference. So my topic is related to mixed ligand complexes with cheap base and 2,2 bipyridine. 2,2 bipyridine. This this compound is very important for our daily life in the sense of medicinal purpose and other activity. Some new ligands metal complexes of transition metal were synthesized from a cheap base and obtained by condensation of isonazide and para-nitrobenzaldehyde as a primary ligand. So we obtained and found one cheap base in his primary ligand, and 2,2 is bipyridine as a secondary ligands. By the combination of these ligands, we obtain cheap base and we synthesize of these compounds in new and modern area. What are the application in our general society for medicinal and modern medical science? So the resultant data exposed to the complexes have a geometry demonstration electrolytic nature. The new compounds and their biological potency were chemically analyzed against E. coli and Bacillus cereus, showing a enhanced activity of complexes against the special as compared to the free ligands. Sir, this is coordination compound and related to chemistry of various ligand with transition metal ion, enhanced by using advanced technique in the field of inorganic chemistry and pharmaceutical industry. Cheap base played an important role as chelating ligand in main group with transition metal ions and gives a stability under suitable condition as oxidative and reductive. The amine group which present in this compound complex and these complexes behave like marginal between hard and soft Lewis bases. Cheap bases obtained from amino and carbonyl group that coordinated with metal ion through azomethane nitrogen. CN linkage which present in our compound of this derivatives imperative role for biological activity like antifungal, antibacterial, anti-cancer, anti-malaria, etc. And it has also catalytic and photochromic properties. Mixed ligand complexes act as a vital role in biological field as enzymic catalyst, and cheap bases derived from heterocyclic compound. This heterocyclic compound like para-nitrobenzaldehyde and furan 2-carbaldehyde. Their complexes are used as a tuberculosis agent. That is very big problem for our world. And this complexes of metal ion and due to its strong redox stability and functionalization. So in this regarding, we do experiment in all used chemicals were purchased commercially from chemical laboratory, and the melting point were determined by using suitable apparatus. Products are characterized by comparison of spectroscopic data like UV visible and FR data, and melting point with authentic sample. The wavelength of absorbance was determined by UV visible spectrometer using a quartz gullet and ethanol as a solution. The magnetic susceptibility of the complexes were measured at room temperature using a balance. And the main general procedure for the synthesis of this compound is by using two ligands. As a first is primary ligands. That primary ligands just I take a name para-nitrobenzaldehyde, and secondary ligand is furan 2-carbaldehyde. By this combination, we prepare cheap base. In this experiment, to the warm methanolic solution and taken 10 ml of methanolic solution as a primary ligand like L1 and warm it of nitrate solution of metal. Just metal use are copper iron, nickel iron were added. After 30 minutes warm in methanoic solution, just like methyl alcohol of 2,2-bipyridine was added drop-wise as a secondary ligand, and resulting mixture was refluxed for about 3 or 4 hours, and the obtained precipitate was filtered, washed, and method and dried under vacuum and dried. Sir, mainly its antimicrobial activity are observed. Those are very useful in our modern medicinal chemistry. These ligands and these compounds were screened for in vitro and antimicrobial activity in DMSO against grammatically E. coli and gram-positive Bacillus cereus stain Kirby or diffusion technique, using basically diffusion technique in this process. A uniform suspension of test organism of 24-hour-old culture was prepared in a test tube containing the sterilized saline solution. Saline nutrient agar was then added in each of the petri dishes. The dishes were related to ensuring the uniform mixing of the microorganism in the agar medium, which was then allowed to solidify. After sterilized, waterman bath filter paper discs were dipped in the solution of each component and placed on labeled plates. The DMSO was as a control of the solvent,
Anilji, we
and
have
so
limitation
we
of time, so
follow
please try to conclude your
suitable
slides.
term.
Anilji,
Sir, please give me few
yes.
minutes, sir.
Okay, try to complete within 2 minutes. Are you online? Raju Ranjit Raj? Sana Samsad, your presentation will begin very soon. So be ready. You have to complete your presentation within 3 minutes. Sana Samsad, you give your presentation.
Am I audible, sir?
Yes, you are audible. Please
Good noon
continue.
to
You are
everyone.
audible.
Good noon to everyone, sir. Myself, Sana Shamshad. I'm from the JP University, Chhapra, under the supervision of Dr. Rita Kumari, and from PG department of the DAV college. My topic is emerging trends in science and technology and its relation to the biomedical waste. I mainly focus my contents about the introduction and antibiotic resistance, pollution created by biomedical waste, and disease arise by biomedical waste. First of all, I want to introduce about the biomedical waste. Any waste arise by the medical facilities such as nursing home, healthcare facilities, medical research centers, clinics, nursing homes are called biomedical waste. Generally, metropolitan cities create about the 30 tons of the biomedical waste each day, and it is a very alarming issue for our present day. So we also and next one is introduction about the antibiotic resistance. What is antibiotic resistance? Waste environment such as unused antibiotic, overuse of antibiotic, uh, and serve as a major source of the antibiotic resistance. Generally, bacteria and fungi are becoming nowadays antibiotic resistance mainly, and it is categorized under the urgent threat, serious threat, concerning threat, and also it is categorized under watch list. First of all, I want to emphasize about the urgent threat, that is Caribbean resistance Acinetobacter, Candid auris, Caribbean resistance Enterobacter, drug resistance Neisseria gonorrhoeae, and serious threat is drug resistant Candida, ESBL producing Enterobacteria, vancomycin bacteria, multi-drug resistance bacteria, drug resistance bacteria. All these are come under the serious threat. And next one is the concerning threat. Erythromycin resistance group such as group A streptococcus is concerning threat, and resistance group B is also under come under the streptococcus. It is also come under concerning threat. Next one is the watch list, that is Aspergillus fumigatus and Mycoplasma genitalium, drug resistance, Bordetella pertussis. All these come and watch a list. First of all, I introduce about the urgent threat, that is Caribbean resistance Acinetobacter. The Caribbean resistance Acinetobacter mainly create the pneumonia and wound, bloodstream, and the urinary tract infection. An estimated case in the present world, according to the data of the 2017, it is 8,500 estimated cases arises, and estimated death is according to the census 2017 is 700. And that it is a fungus and it creates a severe infection And according to the 2018 clinical cases arises is 223. And next is Clostridium Clostridium, that create the diarrhea, colitis, infection of the that is the infection of the colon, and infection per year from the Clostridium official is 2,23,900, and death from these bacteria is 12,800. And the next is disease arised by the waste arises is gastroenteric infection, respiratory infection, ocular infection, genital infection, skin infection, anthrax, meningitis, acquired immunodeficiency syndrome, hemorrhagic fevers, septicemia, bacteriemia, candidemia, viral hepatitis A, B, and C. All these infections and many diseases are arised by the these different type of bacteria and virus, such as enterobacteria, salmonella, mycobacterium, tuberculosis, herpesvirus, Neisseria gonorrhoeae, Bacillus anthrax, Neisseria meningitis, HIV, human immunodeficiency virus. So my topic is antibiotic resistance arised by biomedical waste, and the different disease arised by the biomedical waste, and the pollution created. Mainly pollution is related to earth, soil, water, and this pollution created by the different type of these medical wastes, and the emerging trends in science and technology. Hello.
Try to conclude your presentation within 1 minute.
Yes, sir. I conclude my presentation by saying that the waste, huge amount of the waste, that is 30 ton per year, arised by the present day of the especially medical health care facilities, and the different type of the technologies that is involved in the medical facilities, create a huge amount of the radiation, and that creates a whole environment. So it is a very alarming issue for upcoming generation to think about, to minimize, reduce, and recycle the waste created by the medical facilities. Thank you, sir.
Okay, thank you, Sana.
Hello. Hello.
हाँ बोलिए.
Good afternoon to all of you, respected director, sir, coordinator, co-host, and co-host, sir.
Yes.
Sir, myself, Farah Shakes from Jayaprakash University, Chapra. My topic is a study of birds diversity in Saran district and its conservation. Importance and project and justification for a study. The Indian subcontinent has a great wealth of birds, making it a paradise for the bird watcher. The geographical position, wide altitude range, highly varied climate, and associated diverse vegetation make a suitable place for avifauna in India. The bird's habitat, which can be roughly divided into forest, shrub, wetland, marine, grassland, and agriculture, make them highly diverse species. Birds are a part of the balance of nature. It plays an essential role in the functioning of world's ecosystem in a way that directly impacts human health, economy, and food production. The avifauna of India includes around 1313 species, Grimet et al. In 2011. And according to IUCN Red List in 2016, 84 species of birds are threatened, and India is ranked at the 8th position. Data et al. 2004 reported that a total of 465 species and subspecies of birds in Bihar are commonly found, where 370 species are resident and 140 are migratory. My contents in diversity, habitat, factors affecting diversity, impact of radiation on birds. And first, diversity. You know, the Indian subcontinent has great wealth of birds. Birds are parts of balance of nature. It plays an essential role in the functioning of the world's ecosystem. It directly impacts human health, economy, and food production. Fauna diversity study is an essential ecological tool which acts as an important bioindicator of environmental changes and diversity of any given area. Habitat, forest, shrub, wetland, marine, grassland, agriculture area. Factors affecting diversity: geographical position, wide altitude range, highly varied climate, and associated diverse vegetation. Impact of radiation. Emerging trends in science and technology create more equipment, more radiators, and more radiations has a direct impact upon the decreasing diversity of birds. Many species of birds are in danger due to radiations and other anthropogenic activities. 84 species of birds are threatened. Thank you. Hello. 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 Hello.
Hello, Sana, please conclude your slide. It's already time. We have next participant waiting for their presentation. Hello. इसको ये कर दी ठीक म्यूट कर दीजिए तो इसको सना को इसको म्यूट कर दीजिए सारा यहाँ से ये अपने म्यूट हो गए ठीक है अब नेक्स्ट पार्टिसिपेंट उसका ये निकालिए अविनाश देखिए तो इसमें नहीं आ रहा है, इसमें नहीं आ रहा, इसमें आ रहा है। इसमें ये प्रेजेंटेशन नहीं आ रहा है।
इसमें कैसे जाएगा?
अब जाएगा ना, अब ये तो जुड़ जाना ऑनलाइन।
माइक है सर, इधर बताओ ना।
माइक के पास शुरू हो गया, ऑनलाइन खत्म। ठीक है, 2-3 मिनट में
Dear participant, now we are going to conclude this session, and thanks to you all for your active participation. Almost 350 participants participated in this international conference, and moreover 250 articles we received, and we have published one souvenir, and within a week we are going to publish another souvenir. So the participant whose abstract has been left out, that will be included in the next souvenir. Thank you all. Thanks.
so much thankful to everyone. And special thanks to Science sir. So much thankful to all the delegates, participants, teachers, research scholars, everyone. Thanks a lot. Now we can conclude, sir. Sasank sir, it is over now.